Okay, Priyanka, right?
Yes, sir.
Should I start?
Yes, sir.
Feeling good?
Yes, sir.
Okay. So, Priyanka, just introduce yourself in a brief so we can start further.
Thank you, sir. So, my name is Priyanka. I am from Darbhanga, Bihar. I have completed my schooling from Darbhanga itself, and in 2018, I have completed my undergraduation from agriculture stream from Vishwabharati Shantiniketan, West Bengal. In 2018 itself, I have cleared ICR JRF, through which I got enrolled in molecular biology and biotechnology stream at Indian Agriculture Research Institute, New Delhi. In 2020, I have completed my master's program, and in that year only, I have cleared ICR SRF, through which I got again enrolled at Indian Agriculture Research Institute, New Delhi, for my PhD program. So since 2020, I have been doing my PhD, and I am in the last step of my PhD program where only thesis submission is left. In my PhD, I mainly work on molecular characterization of methylation demethylation dynamics in peach and pea. And
Okay.
my hobbies include cooking.
Okay. So currently you are a research scholar, right?
Yes, sir.
And you are working on the molecular biology and biotechnology. So,
Yes,
what
sir.
is your title of your PhD?
Molecular characterization of methylation demethylation dynamics in PGMP.
Okay. Priyanka, as you have mentioned in your application form that your area of interest is molecular biology as well as transgenic, right? You are working somewhere in
Yes,
the
sir.
improvement of a plant quality in transgenic, right? So,
Right, sir.
tell me, Priyanka, transgenic as well as GMO, genetically modified organisms, are two terms. Are there is any differences, or they are the both these terms are same?
Slight differences is there.
Okay.
Transgenic means transgenic means that a molecule in which some foreign DNA has been inducted. Like that, suppose a plant is there. So in plant, some kind of foreign material like from bacteria or other sources has been included, so that a kind of resistance for pest or for ill improvement can be obtained. Genetically modified or means somewhere some kind of deletion or kind of insertion has been done, so that that molecule, the organism has been modified, but it does not necessarily includes the insertion of the transgenic means foreign DNA molecule.
Priyanka, can we use the
So this is the
statement
basic.
regarding these two terms? That is, all the transgenic organisms considered as a GMO, means genetically modified organism, while all GMO cannot be considered as a transgenic. Can we use this statement?
Yes, yeah, yes, sir. Transgenic would be genetically modified organism, but genetically modified organism necessarily do not need to be a transgenic.
Okay. In India, who regulate the transgenic crop or transgenic plant?
So it is mainly the apex body, Genetically Appraisal Engineering Appraisal Committee. GEAC is the main body under Ministry of Environment and for Environment, Forest and Climate Change, mainly regulate the transgenic in India, the apex body.
Okay. Priyanka, tell me, in molecular biology, each and every aspect and each and every phenomena related to the DNA expressions and ultimately on the protein, right? That
Yes.
means there is a for each and every metabolic processes or phenotypic expressions, there will be a protein for that particular process. Is it possible to alter the expression or function of a particular kind of a protein without changing their gene or gene sequences? Is it possible? My question is that is it possible?
With
Yeah, my question is that is it possible that a function of a protein can be altered or expression of a protein can be altered without going to any interferences or changes in their that gene sequences? Is it possible in molecular biology?
the basic The basics would be that we have to go at the DNA level for changing the protein expression. But with the advancement of technology, I think that some kind of modification in like covalent modification, like protein binding and all those things, may impart some some kind of changes, but the most appropriate would be going to DNA, where we can change any kind of sequence so that the protein expression in terms of changing in amino acid could lead to the changing expression of the protein.
Have you heard about the epigenetic?
Yes, sir. Epigenetics
What is the meaning
is
of
the
epigenetic? Yeah.
Epigenetics is the methylation pattern, DNA methylation patterns, which leads to the changes in the expression of that particular DNA, which is not necessarily to be inherited to the next generation.
Only methylations or some kind also modifications like acetylation, phosphorylation, destylation, dephosphorylations can also be considered as under the epigenetic effect or epigenetics.
No, sir, mainly methylation would be considered in epigenetics. All other like acetylation and all those whatever you have mentioned will be considered as post-translation modification.
Okay, can we consider methylation also as a part of a post-translational modification?
Yes, sir.
Then how
We
you can
can consider methylation.
then how you can
Methylation.
distinguish between post-translational modifications and epigenetic effect? As you earlier mentioned,
Sir,
methylation is a part of a
yes,
epigenetic,
sir.
and also
Yes, sir.
you have replied that phosphorylation, acetylation, or destylation is the part of post-translational modification. So how post-translational modification is differ from epigenetic?
Yes, sir. So in epigenetic modification, we will find the methylation at the bases, the bases present in the DNA. That is cytosine or those mainly the cytosine. We will find that bases which gets attached. Methyl moiety will be attached to that bases, which will ultimately leads to the changes in the sequence and further the expression. But if it is a post-translational modification, then in that case, in the particular amino acid sequence of that protein, suppose for methylation, lysine is the most favorable protein. At that position, methylation, methyl moiety will be attached for its so that its function will get modified accordingly.
Okay, suppose a protein is synthesized in a cell, particular kind of a cells, that is animal cells. Okay, and protein has to be go for the
Yes.
post-translational modifications. So, which different cell organelles will involve in the post-translational modification of that protein?
So mainly the endoplasmic reticulum and Golgi body, in consonance with these two and called as microbodies. These two are the main organelle where protein post-translational modification take place.
Okay, Priyanka, as we all know, there are three domains of life, that is eukarya, prokarya, and archaea, right? And all these
Yes.
three domains of organism have DNA as a genetic material. Okay, so can you justify
Yes,
why
sir.
DNA is a suitable genetic material other than RNA? What is the basic reason and
Yes, sir.
why DNA is considered as more suitable candidate for the genetic material rather than RNA?
Yes, sir. Sir, the main reason for DNA to be a suitable genetic material is its stability and its inherent property to get transferred from one generation to other generation. In case of RNA also, it acts as genetic material in many viruses, but its stability is not that much because of the presence of OH group at position 2 of its ribose sugar, which leads to the means more reactive. RNA is supposed to be more reactive towards the many compounds, but in case of DNA, that OH group is not present, only H is present, so it makes it very stable. So that and that stability is the most important criteria for anything to be transferred from one generation to the other generation. So stability and the inheritance are the two important factors for DNA to be suitable for the genetic material.
Are there any other factor responsible for the stability of a DNA except that is OS present on the present or absence of on the DNA or RNA? Other factors are also.
Sir, one factor would be the presence of cytosine also leads to the some kind of stability, as in uracil present in case of RNA. So, in case of cytosine, the mutation we can find out that this mutation
Cytosine
has
or
taken
thymine?
place. So, oh, I'm so sorry, sir. Thymine,
It's okay.
thymine and uracil. Sorry, sorry, sir.
So okay.
Thymine is the one which is present in. Yes, sir.
Yeah, continue, continue.
Thymine. Thymine is the base which is present in case of in DNA, but this uracil is present in case of RNA. So that uracil imparts some kind of instability to RNA, and in case if any kind of mutation is there in case of DNA, then cytosine will mainly deaminates to changes to uracil. So we can find out that this kind of mutation has happened. So we can tell that this is happening. But in case of RNA, we will not be able to find because uracil is also there and cytosine is also there. So it will change to both uracil only. So there will be no difference in that case. We can find out. So this
Priyanka,
is the one.
Priyanka, have you ever looked the structure of a uracil and thymine?
Yes, sir.
So what is the
Only CH3 methyl group is different. Methyl group
exactly?
is different from
So
in
how
thymine.
a single molecule that is CH3, you are stating that thymine is more stable as compared to the uracil, right? And there is only one group, that is methyl group, is different in thymine and uracil. So how a
Yes, sir.
methyl group is responsible for stability of that base, that is thymine? What is the role of a CH3 group in the stability?
Sir, it will that the molecule will be less reactive because a kind of steric effect will come from the methyl group, which will make the molecule less reactive to other kind of molecule because a kind of it is a bulky group, so that will not allow it to react to many compounds present or molecules present. But if it is not present, then in that molecule will be very reactive, as we can see in case of RNA.
Okay, suppose what will be the possible consequences if genetic material would be suppose is genetic material is RNA rather than DNA? Then what will be the possible consequences on the organisms?
Sir, possible consequence, we can see that in many cases, RNA is genetic material in many viruses.
Yeah.
So there, transcription rate and the replication where mutation is very high. So every means many times it gets changed very rapidly. So kind of stability would be missing in case of if RNA is the main genetic material, and the organism will be changing very frequently the sequences which is present in the genetic material.
Okay, have you heard about the CRISPR-Cas9 system or protein?
Yes, sir.
Okay, can you say something about this?
CRISPR-Cas9. Yes, sir. So CRISPR-Cas9 is a new advancement of the genetic editing mechanism, wherein CRISPR is mainly the short form of cluster regulatory interspersed small short sequences, short palindromic sequences, and Cas9 is an enzyme. So it is mainly seen in case of bacteria that these Cas9 and this CRISPR mechanism is helping in their providing immunity to phages. Phages are those viruses which feeds on the bacteria. So this mechanism has been employed in case of genetic engineering, where CRISPR-Cas9 and a guide RNA. In single guide RNA. In case of bacteria, it is crRNA and transacting RNA that is fused into a single rRNA that is called sgRNA, single guide RNA, and Cas9 enzyme is employed in the genetic engineering mechanism where sgRNA will go and sit at a particular defined position which we want to edit, and then Cas9 has an endonuclease activity, so it will cut at that point. So in that case, we can either delete that point or we can insert something there so that our compound or molecule, whichever is formed, will be genetically edited. So it is mainly employed nowadays for genetically editing as compared to other materials because its off target is very less.
Priyanka, is it possible by using CRISPR-Cas9 technology, can we have a vaccines against HIV in future?
Sorry, sir, I did not. Vaccine? I did not get the whole question.
Vaccines against HIV by using CRISPR-Cas9 technology, can we develop
Yes,
vaccines
sir.
against HIV? As nowadays, till now, we don't have a vaccines for the HIV, right? So can we
Yes,
in the
sir.
future, can we develop vaccines against HIV by using CRISPR-Cas9 technology? Is it possible?
So for that, detailed mechanism has to be worked out. But as in case of HIV, it is a retrovirus. So that mechanism is a bit different there, because it will first convert it into the cDNA, and then copy paste mechanism is operating there. So a detailed mechanism has to be understood first for that CRISPR-Cas to be operative in case of HIV vaccine. But nowadays also mRNA vaccine is coming up. So we can think of as and recently the Nobel Prize has been also awarded for the mRNA vaccine development. So I think we will be, if detailed analysis would be there, then we can go for HIV also. But I'm not sure about it.
Okay, Priyanka, if I asked you how the research community are unable to develop a vaccines against HIV.
Yes, sir. The main problem is that this reverse transcription mechanism, which is operating in HIV, so it is mutating at a very faster pace. Once we are done with one vaccine, the it will create some other form till now. After that development of that vaccine, so that vaccine may not be operative for the new strain which it has been produced due to the reverse transcription mechanism, because in reverse transcription, that reverse transcriptase enzyme, it lacks the proofreading activity, and it can add any amount, means nucleotide, which leads to the high rate of mutation. So this is the main problem which we are facing for HIV vaccine production, that the continuous changing of the genetic material of the HIV due to the mutation.
Okay, recently World Tuberculosis Day was celebrated. How
Yes, sir.
tuberculosis is related to the immunology and what is the molecular mechanism of a tuberculosis infection? How a tuberculosis
So,
infections can be happen in a individual?
sir, whole process I might not recollect at this moment, but it mainly infect the Mycobacterium tuberculosis. That tuberculosis that mainly affect the lungs, and it has a latent period of at least 14 days, and after that it shows up its symptoms, and mainly it progresses in lungs of a human, and then it has 4 stages, that first stage and the second stage, third stage and fourth stage, and accordingly the many that medicines has been come up. But many medicines are there which are which from which the mycobacterium has got resistant. So it is difficult to means control them. So early detection is the one way to control this particular disease. And molecular mechanism I am not able to recollect at this moment.
Okay. In molecular biology, we often use a term mobile element. What is the meaning of a mobile?
Yes. Mobile elements means the mainly used for transposons, which are the parasite DNA, which means mobile means that it can move from one place of a DNA to other place in the other DNA. And it is mainly of two types, class 1 type and class 2 type. Class 1 type is the some mobile element is there, it will cut itself and it will move to a new place and get inserted there. These are called class 1 type, where only cut and paste mechanism is there. But in class 2 type mobile elements, there is a one more thing is there, that it will cut itself, then that RNA will convert it to cDNA, that is called retrohoming. Then that cDNA will get inserted at some places, some DNA where it is required, and this mechanism leads to the copying of copying, then cut and then paste. So there are two mainly, cut paste and copy, cut and paste. So these two are mobile elements because it moves from one position to other position in the
Okay.
genetic in genome.
DNA polymerase is one of the key enzyme used in molecular biology
Yes.
and central dogma process, right? Either for the bacterial cells or eukaryotic cells. So, what are the key fundamental properties of a DNA polymerase?
Yes, sir. So main property is its processivity, that how much DNA per second it is able to add, and at how much point it is able to move to a particular DNA. Then the proofreading activity, that is 3' to 5' exonuclease activity, that is very important for DNA polymerase. And DNA polymerase 3, 1, 2, and 1 both have all three have the 3' to 5' exonuclease activity. Then the addition of the nucleotide per second, that how much nucleotide it is able to add to a growing polypeptide chain. Like DNA polymerase 3 has the 1500 nucleotide addition per second. So these three are the main things which is important for DNA polymerase to be effective.
Okay.
Processivity and nucleotide addition. Yes, sir.
Yeah, you continue. I am just listening to your answer.
Yeah, I was just summarizing processivity, nucleotide addition, and the proofreading activity.
Okay. Uh, in molecular biology, central dogma term is very famous, right? Central dogma process
Yes,
or
sir.
central dogma happens. Why it is known as a central dogma, not simply a process, a molecular biology process? It is considered as a central dogma process. Why?
Because it is the basic of all the things happening in the molecular system, which first initiate with a DNA replication, then it will goes to a RNA transcription, and finally to a protein, which is mainly the expression of a particular phenotype, or in terms of molecular metabolic pathways. So it is the basic of all the metabolic activities going inside a cell. This is the primary step of all the primary step of all the things will happen to next. So this is called the central dogma.
Okay, Priyanka, DNA polymerase is an enzyme, right? And as we know,
Yes.
an enzyme is a very specific for its a particular kind of a substrate. That means a particular kind of an enzyme can only bind to the specific substrate. Okay. So how DNA
Yes, sir.
polymerase can
Yes, sir.
utilize four different kind of a substrate like as ATP, GTP, CTP, and TTP for the polymerization of a DNA strand? How? Are you getting my question?
Exact reason. Yes, sir. I got how it is utilizing the different
Yeah,
substrate.
exactly. If it is an enzyme,
Sir, this is
we consider DNA polymerase in an enzyme, and as we know, each and every enzyme has
yes,
a
sir.
specificity. That means it can only bind to a specific substrate for their activity. So, how DNA polymerase, being an enzyme, utilize four different kind of a substrate for the polymerization of a DNA?
I'm unable to recollect at this moment, sir.
Okay, can DNA
I
poly
have
can
some.
okay, leave it. Can DNA polymerase itself initiate polymerization of DNA?
No, sir.
Then
First, it needs a
then
10
how
to
it
15
polymerize?
days. That for that is used, which will add a 10 to 15 bases at the initial point of the DNA, so that DNA polymerase can sit there to initiate the polymerization activity. So first, it needs the help of RNAs, which will help in synthesis of 10 to 15 base pair. Then only it can initiate the process. Alone, it will not be able to initiate the replication process.
Is there also need of a primer for RNA polymerase?
Yes, sir. Primer is also required.
Primer is also required for the RNA
10 to
polymerase.
15. No, not for RNA polymerase.
Yeah,
I was
I am
thinking
asking
DNA.
about the
No,
RNA
not for
polymerase.
RNA polymerase.
So both
No, sir,
RNA
not
polymerase and
required.
DNA polymerase are an enzyme which have ability to polymerize. Right? DNA polymerase is a
Yes,
DNA
sir.
dependent and it can polymerize DNA strand, while RNA polymerase is RNA dependent and polymerize RNA molecule. So how they are differ
Yes, sir.
from one another, each one in their working mechanism, stating that DNA polymerase require RNA primer while RNA polymerase does not.
Yes, sir. One more difference is that regarding the cofactor they required,
Yeah.
Mg2 plus is mainly required in DNA polymerase, but Zn2 plus is required in case of RNA polymerase. and also RNA polymerase having less processivity and less proofreading activity. They are means around 10 to the power 3 mutation can happen in case of less stable RNA, this much less stable RNA polymerase as compared to DNA polymerase. And also the that proofreading, what I mentioned, that in case of DNA polymerase, it is very means very important for proofreading because it's the main genetic material. So two main thing apart from that primer, in need of a primer, two more thing is that cofactor requirement and then the proofreading activity of the enzymes, where RNA is the less proofreading as compared to DNA polymerase.
Okay. For mutation, generally we consider as a negative factor or a negative phenomena for the individuals or for any kind of organism. Are there any positive impact
Yes.
of mutations in the biological system?
Yes, sir. Positive impact is also
Can we
mainly the
consider
mutation.
for mutation as a positive aspects?
Yes, sir. Mutation are generally recessive things, which is mainly which did not get showed up many times. But there are some gain of function mutations. Those gain of function mutations may lead to means getting some kind of function which is not earlier done by that particular cell or that genetic material. So gain of function mutation is the one positive thing which comes from mutation. But example, I'm just missing. One example was there, but I'm just missing at this moment.
Okay. Uh,
Gain of function mutation.
Priyanka, as you stated that you are a research scholar and currently doing your PhD. Suppose you want to
Yes, sir.
check out the or analyze the protein and DNA binding interaction. So, which kind of a possible
Yes, sir.
technique will you use to find out to check out the interaction of a protein and DNA? Can you name some technique?
Sir, many, yes, sir. Each two hybrids are there in by which we can find out the DNA and protein interaction.
Y2. Y2 is for the protein-protein interactions or DNA and protein. East 2 hybrid system is used for protein-protein interactions or for also protein and DNA.
You are asking DNA and protein, sir.
Yeah.
Okay, so East one hybrid. I was thinking protein, protein. Sorry for that.
Okay.
East one hybrid is there for
Uh,
protein and DNA and protein
any
hybridization.
other?
And sir,
At least,
we
at least,
co-immunoprecipitation.
at least three techniques you have to answer.
Okay, so DNA and protein we can check for chip assay is also there. Chromatin immunoprecipitation is there for where DNA segment and particular protein can we can identify. And then one more is I'm unable to recollect at this moment.
Okay. Have you heard about the food printing?
Yes, sir, footprinting.
What is this?
DNA footprinting is also there. In this case, DNA is digested and then run into a gel. So if a protein gets attached to that DNA, so that portion will not get be restricted because of the presence of that protein get attached to that DNA, particular DNA segment. So it will run slowly because of that heavy molecule. But other DNA where proteins did not get attached, the control sample, we can find that that DNA is getting cut and then running. So this is DNA footprinting where proteins footprint are can be found so that we can know that this sequence is interacting with this particular protein.
Nowadays, in a research, particularly in the molecular biology or biotechnology, qRT-PCR term is used so frequently. As you have read so many papers, such articles as you are research scholars. So qRT-PCR have been used for so many purposes. What is this, and what is the working mechanism of the qRT-PCR?
So qRT-PCR is the quantitative real-time PCR in which it is different from traditional PCR because in traditional PCR, after having that PCR done, we will be able to get that band and then we can come to know that this is the band which we are looking for. But in QRT PCR, in real time, we can get that idea that this is the gene which is getting we wanted that gene, and we don't want that gene. So particularly, it gives the expression of that gene. In the normal PCR, we will may not be able to identify or quantify the expression of that protein. But in case of QRT PCR, quantitative real-time PCR, we can able to find in real time the expression of that gene. While that gene means that mechanism is progressing, we will be able to find that how this gene is expressing itself. And this mechanism is that we generally take cDNA, which we have prepared from the mRNA molecule. We take mRNA molecule as sample, then we prepare it and the cDNA. Then that cDNA is employed for this qRT-PCR mechanism, where specific primers will be used, and one internal control is kept for like ubiquitin or actin for normalization of all those samples which we have taken, and then we can quantify the expression what we are getting after that after the this reaction is over.
Okay, in
So
your
here, no need of going to gel. Yes, sir.
yeah, yeah. Currently you are dealing with the plant improvement, right? So you are working on the plant for genetical
Yes, sir.
purpose. So
Yes, sir.
can you distinguish between the protoplast and protoplasm?
Sir, a protoplast is that when we treat a cell which will lack a cell wall, or means at a place we can remove the cell wall portion of that, and that is protoplast. And the inside material which we are getting, inside material of the fluid, that will be called as protoplasm.
Then what is cytoplasm?
Cytoplasm is the plasma membrane bound material which is inside the cell fluid, where organelles, where organelles is also means we can find organelle in that. And if we remove organelle, then we will be able to tell it is a protoplasm.
Okay, means you are saying that excluding cell organelles from a cells will consider as a protoplasm, while including all the cell organelles will consider as cytoplasm. Is it right?
So in protoplasm, the cell wall will also not be there. Means we have to remove that cell wall. Then only we will be able to term that a particular cell as protoplast or the
Simply if I
fluid
ask
material
what is the
protoplasm.
difference between the cytoplasm and protoplasm of a cell, then how you can distinguish between them?
So cytoplasm will contain cell membrane, which protoplasm will not contain cell membrane.
So is it possible to exist of existence of any cells without cell membrane or plasma membrane? Is it possible?
No, it is not
Then how
possible.
you can say that ki protoplasm is without membrane? Because oftenly we define a living cells having a protoplasm. Suppose if
Yes, sir.
suppose you have a two wooden piece, that is one is the dried and one is the youngest, right? So there, if you determine, if you examine the structure of the cells, there will be no any differences in the dried wooden part and living part of the plant cells, only just of the difference of the protoplasm. So protoplasm is considered as a living
Yes,
part
sir.
of the cells. So how it is different from cytoplasm? Okay, leave it.
Sir, I think
Yeah.
yes, sir.
If you want to say something, then you continue.
Protoplast is mainly that cell membrane. Cell wall has been removed, which we
It's a
use
protoplast, not a
in the
protoplasm.
mainly
If you remove the cell wall from a plant cell, then it will consider as a
yes,
protoplast. It is okay. My question is
protoplasm.
about protoplasm and
Yes, sir.
cytoplasm.
Yes, sir.
Okay, we will
Maybe I am
proceed.
not
Okay,
able to
okay, okay,
tell.
no issue, no issue. Priyanka, suppose you are dealing with the microbes, means bacteria, and you want to examine the morphological structure of a bacteria. There is suppose you are treating with the salinity, impact of a salinity, and you want to examine the morphological changes of the bacterial cells under the salinity impact. So which kind of a microscopic technique would you use for the examination of morphological changes of the bacteria?
Sir, confocal microscopy we can use for morphological changes
Suppose if I
of a
give
bacteria.
two options, that is SIM and TIN, then scanning electron microscope and transmission.
Slim, I think.
Yeah.
So slim, I think, because we are only looking for morphology. We are not cross sectioning in going means the cross section of bacteria. So slim, I think, is
Okay, you
we
you
can
will
use.
go with the SIM, that is scanning electron microscope.
Yeah.
Okay.
Yes, sir.
Okay. Priyanka, electron microscopy is considered as an advanced microscopy other than in comparison to the light microscopy. Is there any difference in working principle, or what make more advanced and more efficient of electron microscope compared to the light microscope?
Sir, mainly the objective resolution. I am recollecting at this moment. Mainly the objective resolution or magnification is more in case of electron microscope as compared to a light microscope.
Okay. Suppose you are trying to express a eukaryotic gene into a prokaryotic cells. So, is it possible
Yes.
to take a simple piece of RNA or DNA molecule and directly insert into the bacterial cells for the expressions analysis, or there will be there will be a need of some modifications prior to insertion of that eukaryotic DNA into the bacterial cells?
Yes, sir. We have to do some kind of modification for eukaryotic gene, because that gene will not be expressing or whatever we want to check any kind of expression in case of prokaryote particularly. And for transfer also, few of the bacteria are naturally recipient, but most of the bacteria are not naturally recipient. So we have to make them recipient also to be able to take that foreign material. So modification will also be required, and residency of that particular bacteria or prokaryotics is also required.
Okay. Have you heard about the C value paradox?
Yes, sir.
What state that? What is the meaning of
So
C value paradox? Yeah.
C value, yes, sir. C value paradox is the complexity level of an organism and a gene size of an organism. Suppose
Okay.
a organism is there whose gene size is 16 GB, but and one organism is there whose gene size is only 4 GB. For 4 GB, the genome is maybe more complex as compared to the 16 GB. So the paradox between the complexity and the gene size is the C value paradox.
It's called C value. What is the meaning of a paradox?
C value enigma and paradox is that the different means the not matching of the gene size and the gene complexity level.
Okay, got it. So Priyanka, now we will
Yes.
ask some another questions, but currently, uh, right now you did well, and my question is over from biotechnology or molecular biology. So अभी अगर आपके पास कुछ आपकी तरफ से सवाल है जो कि आपके मन में है और आप पूछना चाहते हैं, तो आप उसको पूछ सकते हैं। देन इसके बाद आपको हम सजेस्ट करेंगे क्या क्या चीज़ों पे आपको थोड़ा सा और फोकस करने की ज़रूरत है। ओके, सो नाउ इट्स योर टर्न, वॉट एवर यू वॉन्ट टू
Yes, sir.
आस्क, या यू कैन। आप
सर,
रिलेटेड, हाँ
पहला
आप,
तो ये कि
हाँ हाँ पूछिए पूछिए आप।
पहला क्वेश्चन ये है कि वहाँ पे मतलब अगर जैसे कि बायोटेक में बहुत सारे एलिजिबल ब्रांचेस थे लाइक माइक्रोबायोलॉजी एंड ऑल दोस, सो हमें ये चांस मिलेगा कि वहाँ पे बोलें कि हाँ आपका कुछ ऐसा पर्टिकुलर टॉपिक है जो कि मतलब आप बोलना चाहते हैं, या आप उससे हम आपको क्वेश्चन पूछें, ऐसा कुछ मतलब
डेफिनेटली
चांसेस दिए
डेफिनेटली मिलेगा
जाएंगे?
प्रियंका।
ये एक क्वेश्चन था
या
मेरा।
डेफिनेटली डेफिनेटली मिलेगा, बिकॉज इंटरव्यूअर जो होते हैं वो पहले से कोई क्वेश्चंस फ्रेम करके नहीं आते हैं। वो आपसे जब इंटरेक्ट होते हैं तो आपके ही रिप्लाई से ही वो सवाल को जनरेट करते हैं। और ये क्वेश्चंस आपसे ज़रूर ऑफ़र किया जाएगा कि आपका फ़ेवरेट टॉपिक क्या है? आप किस टॉपिक पे ज़्यादा कम्फ़र्ट हैं या करेंटली आपका डोमेन क्या है? जब वो आपसे ये सवाल पूछते हैं दैट मीन्स वो आपको ऑफ़र देते हैं कि आप इंटरव्यू को अपने हिसाब से आगे कैरी फॉरवर्ड कर सकें, राइट? क्योंकि जब आप कोई एक स्पेसिफिक एक्चुअली biotechnology is not a simple term and is not a single area. It's a huge number of diversity. बहुत सारी डाइवर्सिटी है इसमें। माइक्रोबियल बायोटेक्नोलॉजी है, एनवायरमेंटल बायोटेक्नोलॉजी है, प्लांट बायोटेक्नोलॉजी है, एग्रीकल्चर बायो, बहुत सारे हैं। तो आपसे ये सवाल पूछा जरूर जाएगा, आपसे ये क्वेश्चन जरूर ऑफर किए जाएंगे कि आपका जो स्पेसिफिक एरिया है या आपका फेवरेट जो टॉपिक है वो क्या है। एट दैट टाइम आपको इस चीज़ को लेकर के थोड़ा सा पहले से ही प्रिपेयर होना है कि सपोज जैसे अभी आपने ये फॉर्म में फिल किया हुआ था कि आपका मॉलिक्यूलर बायोलॉजी और बायोटेक्नोलॉजी फेवरेट टॉपिक है, राइट? और आपने आंसर भी बहुत अच्छे से दिया। अभी उस पर भी हम लोग आएंगे, डिस्कस करेंगे। तो ये सवाल आपसे पूछा जाएगा डेफिनेटली, और आप उसके लिए प्रिपेयर रहिए। जो भी आपको लगता है कि सब्जेक्ट पूरे बायोटेक्नोलॉजी का लाइक एज मॉलिक्यूलर बायोलॉजी, बायोकेमिस्ट्री, बायोटेक्नोलॉजी, इम्यूनोलॉजी, वॉट एवर, उस टॉपिक को आप वहाँ पे अच्छे से बताइए ताकि वो फर्दर उससे आपसे सवाल कर
Yes, sir.
सकें। और ये जो आपका इंटरव्यू होने जा रहा है यहाँ पे मैक्सिमम जो क्वेश्चंस आपसे पूछे जाएंगे वो आपके ही डोमेन से पूछे जाएंगे, क्योंकि उनको ये पता है कि आपको आईपीआर का बहुत ज़्यादा आइडिया नहीं है। आपने अपने एकेडमिक में कहीं पे भी आईपीआर को स्टडी नहीं
Yes.
किया हुआ है। तो अगर आप आईपीआर को लेकर के थोड़े नर्वस
Yes,
हैं
sir.
तो आपको नर्वस होने की ज़रूरत नहीं है। आपको थोड़ा बहुत जो आइडिया है वो ठीक है।
Yes, sir.
आपका मेन जो फोकस होना चाहिए आपके अपने फील्ड पे, दैट मीन्स सब्जेक्ट पे, और स्पेशली उस पे ज़रूर होना चाहिए
Yes, sir.
जिस चीज़ को आप वहाँ पे स्टेट करते हैं कि सर ये वाला मेरा टॉपिक फेवरेट है और इस पे हम कंफर्ट हैं।
Yes, sir.
ओके, सो नियरली
Yes, sir.
15 to 20 मिनट्स आपको टाइम दिया जाएगा और आपसे कन्वर्सेशन की जाएगी ऐसे ही
Yes,
जैसे सवाल यहाँ पे आपसे पूछे गए हैं। तो अगर आप इस टॉपिक पे कंफर्ट रहेंगे
yes,
तो
sir.
अच्छा होगा। और कुछ सवाल है
Yes,
आपका?
sir. और मेरा सेकंड क्वेश्चन वही था जो आपने अभी आंसर कर दिया कि आईपीआर का कितना मतलब लेके चलें कि इतना कुछ पढ़ना है
आपको
आईपीआर में
आपको
अगर
बर्डन लेने की
कुछ
ज़रूरत
भी
नहीं है।
इम्पोर्टेंट
अब
है।
नेक्स्ट मॉर्निंग आपका इंटरव्यू है तो आपको अननेसेसरी रात में बहुत पैनिक होने की
यस
ज़रूरत नहीं है। आप वैसे भी 4 घंटे में आईपीआर में मास्टर नहीं हो पाएँगे और ना ही आप उसको पढ़ पाएँगे, है ना? तो अननेसेसरी क्या अपने ब्रेन पे और अपने माइंड पे प्रेशर दे करके जो चीज़ें आपको आती हैं और जिसमें आप कम्फर्ट हैं उसको भी डिस्टर्ब करें, उसका कोई बेनिफिट नहीं है, ठीक है? जिन क्वेश्चंस का आंसर आपको इंटरव्यू में नहीं आएगा, आप बहुत ही पोलाइटली उसे डिनाई कर देना कि सॉरी सर, राइट नाउ आई एम अनेबल टू स्पीक समथिंग अबाउट दैट, राइट? आई डोंट हैव आइडिया। सो ये
यस
ऑब्वियस सी बात है कि इंटरव्यू में जितने भी सवाल आपसे किए जाएंगे उनमें से कुछ क्वेश्चंस का आंसर आपको नहीं पता होगा, है ना? तो
यस
ये ऑब्वियस है, तो उसको लेकर के आपको बहुत ज़्यादा पजल होने की ज़रूरत नहीं है। आईपीआर आपका तैयार नहीं है तो आपको उसके लिए एकदम परेशान नहीं होना है। आप अपने सब्जेक्ट पे अच्छे से पकड़ बना के रखिए और उम्मीद की जाती है और आपसे एक्सपेक्टेशंस ये रखी जाएगी कि हाँ, आपका मतलब जो इंटेलेक्चुअल है और आपका जो आंसर्स है, आपके गिवन स्पेसिफिक एरिया पे थोड़ा-सा रिप्लाई निकल कर के आए। ये ज़्यादा इम्पोर्टेंट है। अगर आप
यस यस
खुद को अगर सैटिस्फाई कर पाती हैं देन डेफिनिटली आपका इंटरव्यू बहुत अच्छा होगा, उसमें कोई डाउट नहीं है। और नेक्स्ट कोई सवाल है आपका?
यस नहीं सर अभी और नहीं।
अच्छा, ओके। सो प्रियंका, आपका इंट्रोडक्शंस का पार्ट अच्छा है, कोई प्रॉब्लम नहीं है उसमें। आपने जिस तरीके से कॉन्फिडेंटली इनिशियली स्टार्टिंग से ही आप कॉन्फिडेंट दिखी, मतलब नर्वसनेस आपके फेस पे नहीं दिखा। एंड होपफुली कि यही कॉन्फिडेंस आपका वहाँ इंटरव्यू में भी बना रहे और आप अच्छे से परफॉर्म करें। बाकी ड्रेसिंग सेंस आपकी अच्छी है, ऐसे ही चूंकि गर्मी का मौसम है और आपको बहुत ही सिंपल जिस तरीके से आप हैं, मैं मतलब कहूँगा कि ऐसे ही आप चले जाइए इंटरव्यू में तो ज़्यादा बेटर बेटर होगा, ठीक है? तो आपका जो ड्रेसिंग
यस
सेंस है वो अच्छा है, उसमें कोई दिक्कत नहीं है। बाकी आपकी सब्जेक्ट पे पकड़ बहुत अच्छी है, नो डाउट, डेफिनेटली।
थैंक
नियरली
यू
मैंने 15 से
थैंक यू
18 क्वेश्चंस मैंने पूछे होंगे आपसे और अलग-अलग टॉपिक से पूछे होंगे, करंट का सिनेरियो से लेकर के और इन डेप्थ में भी जा कर के। बट ऐसा मुझे लगा कि सिर्फ एक क्वेश्चन आपको थोड़ा परेशान किया, दैट इज़ द प्रोटोप्लाज्म एंड साइटोप्लाज्म। इसके अलावा आपने
यस
सारे क्वेश्चंस का आंसर बहुत ही अच्छे से दिया है। उसमें कोई डाउट नहीं है।
थैंक यू
तो
सर।
आपके पीएचडी करने का बेनिफिट आपको है, और आप है ना, तो उसमें आपको दिक्कत नहीं है। अगर
थैंक
आप
यू।
आज रात में कुछ नहीं भी पढ़ेंगे, आप सिर्फ अपने आप को रिलैक्स कीजिए और जो भी चीज़ें आपके मन में है उसको रिकॉल करते रहिए, तो कोई प्रॉब्लम आपको नहीं होने वाली है। ठीक है, लैंग्वेज आपका ठीक है, आप एकदम फ्लुएंट है, कम्युनिकेशन में कोई दिक्कत नहीं है। आप इस तरीके से ही, क्योंकि वहाँ पे इंटरव्यू ओनली इंग्लिश मीडियम ही होगा, जैसा अभी हमारे आपके बीच में बातें हो रही है, ठीक है, तो उसमें भी कोई आपको दिक्कत नहीं है। तो मिला जुला करके ये है कि आपको कुछ करने की जरूरत नहीं है, आप एकदम रिलैक्स रहिए, अच्छे तरीके से कामली आप आज नाइट में अच्छे से सोइए, ताकि कल आपका माइंड एकदम एक्टिव रहे और आप अच्छे से परफॉर्म कर पाए। कितने बजे है आपका इंटरव्यू?
मेरा रिपोर्टिंग टाइम 1:30 है।
ओके, 1:30। और अभी आप जहाँ स्टे की हुई हैं वहाँ से कितना दूरी पे है आपका इंटरव्यू का जो वेन्यू है?
हाँ, वो जगह तो दूर है। मैं अभी पूसा में हूँ, जहाँ पे मेरा पीएचडी था। तो मैं कल रात को ही आई थी, तो मैं अभी पूसा में हूँ,
अच्छा।
राजेंद्र प्लेस। पर वो अभी ओखला है, तो 21 किलोमीटर्स है यहाँ पे। तो
ओके।
मेरा प्लान है मैं 10 बजे तक निकलने का,
हाँ, कोशिश
उसके समझ
कीजिएगा
से।
कि टाइम बिफोर निकल जाइएगा, ऐसा ना हो कि एकदम नाप तोल के टाइम लेके चलें,
हम्म,
बिकॉज ट्रैफिक का
हाँ
कोई
हाँ
कोई सर्टेनिटी नहीं है ना। तो आपके पास अगर टाइमिंग
हाँ
है, तो
हाँ
आप,
हाँ।
गर्मी का टाइम है, 9 बजे 9:30 बजे तक आप आराम से रूम को लीव कर दीजिए, निकल जाइए। वहां जाकर के आप रिलैक्स करेंगे, आप बहुत कॉन्फिडेंट रहेंगे। पता लगा कि आपका 1 बजे एंट्री टाइम है और आप पौने 1 बजे पहुंच रहे हैं, तो फिर आप इतना घबराए हुए से रहेंगे ना कि
हम्म,
आपको अपने आप में रिलैक्स करने में बहुत टाइम लग जाएगा।
हम्म,
तो एयरपोर्ट वाले रूल को फॉलो कीजिए कि 1 बजे की फ्लाइट होगी तो 2 घंटे पहले ही एंट्री करा लेते हैं, ताकि आप वहाँ रिलैक्स हो जाते हैं, है ना।
हम्म,
उनका मेन पर्पज यही होता है कि जो भी
हम्म,
बंदा आया है घबराया हुआ ना रहे, रिलैक्स माइंड में रहे, तब जाकर के उनको फ्लाइट में
हम्म,
सीटिंग कराया जाए। तो आप टाइम से निकल जाइए प्रियंका, और बाकी और कोई चीज़ की दिक्कत नहीं है, आपका इंटरव्यू बहुत अच्छा होगा, हमारी तरफ से आपको ढेर सारी बेस्ट विशेष। और
थैंक
ज़रूर
यू
कल
सर।
आप इंटरव्यू देकर के आइए,
थैंक
तो
यू
अपने
सर।
एक्सपीरियंसेस को शेयर कीजिए, ग्रुप बना हुआ है, आप उस ग्रुप की मेंबर हैं।
जरूर।
ओके। तो मेरी
जरूर
तरफ से
सर।
आपको सजेशन तो
यस
नहीं
सर।
कहूँगा, बस सिर्फ यही है कि आप जैसे हैं, आपका प्रिपरेशन अच्छा है, कोई आपको बड़ा लेने की जरूरत नहीं है, ठीक है, आप एकदम रिलैक्स होकर के कल इंटरव्यू देके जाइए, एंड होपफुली कि 50
थैंक यू
सीट में
सर।
से एक सीट आपका भी हो।
22 में से।
अच्छा, ओके। आपने बाइफरकेट कर दिया उसको कैटेगरी वाइज।
हाँ
चलिए,
हाँ।
ओके, ठीक है प्रियंका, और कुछ?
थैंक यू सर।
चलिए,
नो सर,
वंस अगेन
थैंक यू,
विश यू
बहुत
ऑल द
अच्छा
बेस्ट फॉर योर
एक्सपीरियंस रहा
इंटरव्यू,
था।
एंड
थैंक यू सर।
ओके, गुड नाइट।
थैंक यू सर।